I would like to welcome all our incredible audience that is joining us today from all our platforms, Zoom, YouTube, and Facebook, which we are making this streaming. Under the framework of the Science Outreach Project led by Dr. Natalia Diaz, researcher of the Scientific and Technological Bioresearch Nucleus within the organizing team, promote this webinar CRISPR, a next-generation molecular tool, with the aim of communicate CRISPR technologies and their potential application associated to different areas. On this occasion, I would like to introduce Dr. Holger Puchta, the director of the Botanical Institute, who holds since 2002 the chair of plant molecular biology and biochemistry at the KIT in Germany. He was worldwide the first scientist to demonstrate the site-specific nucleases that apply to induce different kinds of controlled change in plant genomes. His group elucidated major mechanisms of DNA double-strand break repair, and he was one of the leading scientists adopting the CRISPR-Cas technology to plants. This year, his group was able to achieve another important breakthrough for breeding, CRISPR-Cas-mediated plant chromosome engineering. For his work on plant genome engineering, he was named Pioneer of the Plant Biotechnology by Plant Technology Journal, and he was awarded twice with an advanced grant of the European Research Council. Today, he will share with all of us the talk entitled Applying CRISPR-Cas to Plants, from gene editing to chromosome engineering. Very good afternoon, Dr. Holger. First of all, thank you very much for accepting this invitation. It is a pleasure to have you here today. The audience is completely yours.
Well, first of all, thank you so much for inviting me. I mean, of course, I would have loved to come in person to your lovely country. I have to say, I have been visiting Chile before. It's really lovely, and I liked it very much. But I'm also, of course, honored to be invited and give a talk. And as there is no other way, it's also fine, of course, to do it online. So in principle, I will tell you a little bit about different aspects of CRISPR-Cas use in plants. And to better understand this, I first want to dig deep in the history of plant breeding, telling you something about how gene editing evolved. And then I will also go in some further, more sophisticated applications of genome engineering. This is gene targeting, changing the genome by homologous recombination, because many people that are working with plants are interested in establishing these technologies in their labs. And then, as was already mentioned before, we recently also for the first time succeeded to engineer chromosomes in plants. And of course, this will be a very important third part of my talk. So I have to get used to switching the slides. So first of all, let's deep dig into history and into plant breeding, because maybe at your university, people know well what is plant breeding all about, but this is not always the case when I give these talks. So in principle, what is breeding all about? So breeding is all about to make out of something that looks like a plant, some very strange kinds of monsters that are very nice food for humans. What you see here is an example, a brassica plant. And out of this brassica plant, breeders were able to develop Brussels sprouts, broccoli, cauliflower, cabbage. And during this process, there was a lot of genetic variability in brassica, in the original plant, and this enabled to have all these different kinds of plants. But this genetic variability during breeding in general, and this not only applies for the vegetables shown here, is lost. And therefore, it's getting harder and harder to further improve this plant, because the breeder has no more genetic variability to use. One way, of course, is to cross in genetic variability, but this is also very difficult. And therefore, quite a long time ago, there was this development to try to introduce genetic variability in a plant by a totally different mean. And the mean was just to induce damage to the genetic information. And because damage is induced, this damage has to be repaired. And the repair product in the end is different than the genetic information was before. And so in principle, this is a basic principle also that was evolved in classical breeding. We are inducing by brute force in what people did at this time, by inducing with X-rays, for example, breaks in the genome. So it's the same principle we are using today also with the CRISPR-Cas system. And then these breaks are repaired. And what you see here is a way how breaks are repaired. So you see here, you induce a break in a double-stranded DNA molecule, then it's repaired. Sometimes it's repaired in a way that sequences are lost during the repair process, and then the genetic information is changed. So you might ask yourself, well, in principle, does this at all make sense if it comes to breeding? Because all you can do is you can destroy genes, you can knock out genes. But you have to think about what is important for breeding, and it might well make sense to knock out specific functions because the specific functions you don't want to have in the field. I want to give you a very simple example, and this is the stemness, long stemness, short stemness of cereals. A cereal in the wild, in any case, needs very long stems to grow fast, to be close to the sun, to be the first in competition. But if you have very long, thin stems in a field and a storm comes, these stems will not hold the storm, but the whole field will be destroyed, and the yield for the farmer is lost. So in principle, what you would need are short stems. And indeed, if you knock out or destroy this genetic information that is responsible for long stems, you get stouted plants with short stems. And this is the way how this thing really functions. And in principle, this methodology was developed more than 70 years ago. So since in the middle of the 20th centuries, plants were either treated with chemicals or treated with radioactivity. And of course, this resulted in many different mutations all over the genome. And most of the mutations were not interesting for breeding. So there were a lot of adverse mutations, and you have, of course, to get rid of them again, and you have to cross them out. And therefore, this procedure is successful. It takes a very long time, and you never can get rid of all these mutations. So you still have in the genome a lot of further unwanted mutations, what we also call off-site mutations. But nevertheless, this is tremendously successful all over the globe. So maybe today you had already a grapefruit. Most probably this grapefruit looked like this. So it was seedless. And in principle, these plants were all produced by this technology. Or maybe one of you will have tonight. I mean, here it's already evening, but I know that you still have some hours more to go before it's getting dark in Chile. If you have a beer tonight, and this beer comes from short-stemmed barley, most probably this is also produced with this technology. So all in all, worldwide, 3,000 cultivars are grown at the moment, and they all rely on this technology of induced change in an unspecific way. So, of course, this technology is successful, but it could, of course, be much better, much faster. And therefore, since a very long time, there was a question, can we improve this? And if you want to improve things, and it's all about biology, you of course have to look, what is biology doing? And in biology, you have this problem once and again during the lifetime also of a human, for example, to produce antibodies, also in inheritance, to combine genetic information in a new way. And for combining this, you have to cut this information. So there are enzymes around that are cutting genetic information in all different kinds of organisms, and to really test this and check whether we can use these enzymes for reading, one has to first, and this was, of course, history in the last millennium, has to find enzymes that are really specific enough that they only cut once in the plant genome. And I was very lucky, and this was already mentioned, that we were really the first who had an enzyme in hand that people discovered before. In yeast cells, this was this enzyme, it was called IAC1. And this enzyme was specific enough. It had an 80-mer recognition site. It was specific enough to cut only at one site in a plant genome. And this gave us the possibility at this time, this was in 1993, to learn more about what can really happen if we induce a single break by an enzyme. And therefore, we have a quite long experience inducing breaks in plant genomes. And I just want to mention a few things which we found very long ago, but which are also very relevant if it comes to using the technology in 2021. So on one side, we found, and this will be interesting also later for the talk, that if you induce breaks in somatic cells of these plants, there are in principle two different pathways that if you look at them, differ a little bit how these ends can be rejoined. So the rejoining of broken ends is very efficient in somatic cells. It's different to meiotic cells where you have homologous recombination. I come to this later. But in somatic cells, the ends are just put together that no genetic information is lost. And what you can see here is this classical end joining pathway. The ends just come together. And if there's one or two nucleotides lost and you have an open reading frame, you, of course, induce a knockout. And on the other side, you have this alternative non-homologous end joining. It's also called microhomology-mediated end joining, where the ends are chopped off to a certain time, one strand each, and then microhomologies find each other and base pair, and then bigger deletions are formed. And because there are bigger deletions, this pathway is even more mutagenic. And this we already analyzed 20 years ago. And another very interesting thing we found all these years ago with the IAC1 enzyme was that we were able, when we induced a break, also to find the DNA is integrating into the break. So this was the first indication. We used tDNA. This was the first indication all these years ago that tDNA integration is in principle relying on the cellular machinery of the plant, and the integration occurs at double-strand breaks. But this also should show you, and that's why I mentioned it to you, is you always have to be aware if you induce a double-strand break. In most of the cases, it's just rejoined, but in some cases, many other different things can happen. And one of the things is that there are other sequences that can integrate into the specific site. And I also tell you this because there was once and again concerns raised when the CRISPR-Cas technology was introduced into different experimental systems, and people found things like that. They always thought this is because of CRISPR-Cas. No. This is because of the natural repair mechanisms that occur during break repair, in this case in plants, but a similar mechanism also exists, of course, in mammals. Okay, the problem with these early enzymes we used 20 years ago was that we just could cut one specific site in the genome. And of course, if you are interested in applications, but you want to induce changes in specific genes you're interested in the genome, you cannot use this tool. So we saw a longer development of artificial nucleases. This was the zinc finger nucleases. They were applied first in plants in 2004, and in 2010 transcription activator nucleases. And both kinds of nucleases in principle were already prone of directing breaks to specific positions in the genome, and they did this by DNA binding domains that were from transcription factors, and you could program the specificity of these transcription factors in a way that enabled you to induce breaks. So the technology to induce specific breaks exists much longer than CRISPR-Cas was discovered. But CRISPR-Cas, as you all know very well, was discovered in 2012. And obviously, this was a big revolution, and it also led, as shown here, that Emmanuel Chapoutier and Jennifer Doudna got the Nobel Prize in 2020 for this discovery. The question is, of course, why was this technology so much better than anything else? Well, what you see here is this Cas9 enzyme as a specificity in respect to all other enzymes is here defined by an RNA molecule, a guide RNA molecule, and not by a protein domain. And this makes the system so much better than the systems that have been used before, because you have to imagine to construct a protein to select for specific bindings. This took half a year, 3/4 of a year, and then you had to pay $25,000 to get an enzyme. And in the end, because we used all this, I can tell you the cutting was not very efficient. And sorry, is there any? I hear voices. Is there everything fine with my presentation? Yes. Okay. So in principle, here, what we have is we can change the RNA overnight by simple cloning, and we get for €25 in 1 day or in 2 days a new enzyme. So this makes the system so much better. So it's much faster. The Cas nucleases are cutting very efficiently, much better than the zinc fingers, for example. And the second very important difference is that you can use several of these guide RNAs at the same time in a cell. So this means that in principle, you can induce not only one break at a time, but several breaks by using several guide RNAs. And that's why the system is so much better. And that's why the system really came over biology like a tsunami, and it changed everything. It also changed completely my scientific life, I have to say, and of many others. And we are now really able to induce mutations in all kinds of organisms where we can bring these enzymes in. And this is a tremendous difference to what biology has been before this time. Okay, so in principle, we started to work with this soon after the first publication was published. I had a very motivated PhD student, Frieda Fauser. And at the time when we started, nobody was really able to induce mutations into Arabidopsis, which you all know is a model plant, a very important model plant for plant biologists. And so we set up a system that we can transform into Arabidopsis. You might know that Arabidopsis is transformed via this Excel transformation protocol where we transformed the open reading frame of the Cas nuclease and the guide in. And we used the UBI promoter. Before, people used 35S promoters, which is the most common promoters. If it comes to plant biology, we used the ubiquitin promoter. And indeed, we immediately were able to get stable mutations. And these stable mutations could then be transferred to the next generation. I think this is, of course, very important if you want to work with mutants. And we were in the beginning obtaining mutants at a frequency of 10 to 20%. And this is fine for most applications. But as you will see later on, we are interested in very specific changes in the genome that occur relatively rare. And therefore, since this time over the last years, we tried to characterize or improve other CRISPR-Cas nucleases that might even cut more efficiently. So Janet Steiner, another PhD student in my group some years ago, could show, at least for Arabidopsis, that the Cas9 nuclease of aureus, which is quite closely related to the pyrogenase enzyme in structure, is much better in cutting. So you see here we have a cutting over 90%, which really gave me at this time a positive heart attack when I saw this for the first time. And so, as you will see later, we are at the moment applying for most purposes this enzyme, but in the last years, a second class of enzymes was characterized besides Cas9, and these are the Cas12a enzymes that are also now widely used in different organisms. As a problem with the Cas12 enzymes for plants is that they are quite temperature sensitive. So this is they are coming from a human pathogen. The optimum of activity is 37 degrees. So you really, if you think about plants being grown at 20 degrees, these enzymes might run into trouble. And therefore, we try to optimize these enzymes to get an enzyme that is working better in plants at lower temperatures. And we exchange several amino acids in the open reading frame. Other people made structures of this, and so we just used their data from other Cas12, and so we introduced, besides other things, when I say we, I of course mean my PhD student, Patrick Schinderle, introduced into this a single point mutation. Of course, he tested more things, but important is only the single point mutation. And with the single point mutations, we suddenly see that we get a very efficient induction of mutations. This is our readout, of course, because we don't measure directly double-strand breaks. And what you see here is in blue, the wild type CAS and our temperature tolerant CAS. And here's the activity from 0 to 100% at certain sites. And you see this enzyme where you have here 10 to 20% cutting. You have here up to 90% cutting. And this is applicable for many different sites. And so this is indeed, if you would ask me if you're interested in applying CRISPR-Cas enzymes to different species, this is really for me at this very moment the best enzyme we have in the hand. We also tested it recently, and this will be published soon in Plant Biotechnology Journal in tobacco, and also in tobacco, as you see here, it's better than the classical LB12. So if you're interested in using this enzyme for your specific research purposes, I'm very happy to hand it out freely to everybody, even without MTA. You just have to write me an email, and then we can send you the enzyme, and you can try in your favorite plant to induce mutations with it. So in principle, I want to make, besides this historical introduction, I also want to make a short deviation to politics, because I think this is very important. It's also important, and I'm not sure how familiar you are with this situation in Europe, and I just want to mention this. So if you look at these plants, double-strand break repair is a natural process. If there is no transgene integrated, then in principle, these plants cannot be discriminated from natural occurring plants. We have a lot of experience with molecular scissors in more than 25 years, and we hardly produce any off-targets in contrast to the classical mutagenesis protocols where you have hundreds and thousands of off-sites. So in principle, these plants should be grown everywhere, and there should be no single risk growing these plants in comparison to normal wild-type plants. But unfortunately, in Europe, the situation is not like that. So in 2018, there was a ruling of the European Court of Justice that all these plants that are produced with CRISPR-Cas, even if they don't contain any transgene, they have to be regulated as GMOs, as plants that contain transgenes. And regulation in Europe means that you have to go through a procedure that takes 10 years and costs 20 million euros before you can bring out these plants on the field. And the only exception the judge has made is for classically mutagenized plants, what I mentioned in the beginning, because they are so long now in use that the judges regard them as safe. So in principle, for us scientists, this is, of course, a really ridiculous situation. Think about a classically mutagenized plant. there you have at least, if you just take an example as barley, as I do it here, you have at least 10,000 offsites mutations in the field in every single plant due to this classical mutagenesis. And everybody is happy with this. So if you think about mutation frequency, so the question, if you have two plants in the same field, are they really the same? And I tell you, no, they are not. Because in barley, for example, two plants in the same field, even if they are very closely related, there are 100 natural mutations occurring with every new generation. So there is mutation occurring in the field all the time. And then we come with our molecular scissors and make a single mutation. Maybe there are one or two offsites. This is noise. This is beyond the genetic noise. So this is so much better than anything else that was developed till now, that I always say that with CRISPR-Cas we are better. We are better than nature. And so there's a long debate going on since 2018 in Europe to change the situation, because it has also very drastic consequences also to breeders and farmers worldwide. We as European scientists fight hard for a change. So I was member of a committee of the German Academy of Science, and we also met the German ministers for agriculture, the German minister for science, which in principle agree with us. But of course, as you know, politics is complicated. There are many different parties. It's hard to convince everybody. But I'm still hoping that there might be a change of situation in Europe in the next years, because this would also be beneficial for the rest of the world, because then it would be very easy to also import these kinds of crops into the European Union, also from South America, for example. Okay, so far to politics. Now I come to gene targeting, because I know that many people are interested to really, in a controlled way, change specific sequences in the genome. And this you can only do by homologous recombination, because if you induce just a simple break, you get different kinds of mutation. And we could show all these years ago, in the last millennium already, if you induce a break, you can get specific integration by homologous recombination. So you have here a tDNA that has homology at these ends. And if you just try to integrate this tDNA in this homologous region without inducing a break, you have to transform this tDNA 100,000 times. So this is happening very seldom or not at all. But we could show if we induce a break, then we can dramatically increase the frequencies to up to 1 out of 100. So in 1%, about 1% of the cases, you get homologous integration. And therefore, this is now also a tool to change this plant genomes in a more controlled, specific way. I have to say the downside is that a lot of efforts have been done since the last millennium to really increase this 1%, and we are a bit better now, as I also will tell you, but still, it's not the main pathway, of course. In somatic cells, you change somatic cells, the main pathway is non-homologous end joining, and you have to try your best to find these homologous recombination events. And because it's hard to find these events and you have to transform a lot of these things, we developed a technology which we call implanter gene targeting. And the idea behind this technology was just to say, okay, if it's so much work, especially if you have plants that are hard to transform, to try to make all these transformations, it's not the technician or the students that should do the work, but it's the plants that should do the work. So we bring in the transgene before into the genome, and then we induce a break, but we not only induce a break at the site where we want to integrate the DNA, but also we cut and activate out the vector sequences. This is shown here. So this is the process. We use the enzyme. This can be ISC1 in the beginning or later on CRISPR-Cas, and then we induce, and homologous recombination takes place between the sequences which have the same color, and the donor is fused, and you can segregate the donor out. And in principle, you have what you want. And the nice thing is you just analyze seeds. So there is no tissue culture necessary for this, because you just go for the seeds and check in which of the seeds is the correct targeting event. And to work with this and to improve this, we used in Arabidopsis, which is our model plant, as I told you before, we used the ALS gene, because if we induce a point mutation, a directed point mutation in this gene, and this was work done by my excellent PhD student, Felix Wolter, we are able to select for the result, because these plants are then resistant to herbicides, and you can grow these plants on herbicide-resistant plates, and then you can, in principle, select for this, and you have a very, very nice readout. And using these technologies, we tested how can we be more efficient. And what we found out is that if you use aureus Cas9, which we knew from before, it's a better cutter, and also use specific expression of the enzyme, not everywhere in the plant, but only in the egg cell, which is in any case a guarantee that this will be transferred to the next generation, this change. So if you use egg cell specific expression and a good cutter like aureus Cas9, we can enhance the frequencies. And what you here see are different Arabidopsis lines, and this is the respective control where we don't use the egg cell promoter. And you see now for specific lines, we can go up to 5-6%, and we have in many lines, we have at least 2% activity. So this was already a good thing. And as I mentioned before, we also developed this Cas12 enzyme recently. And the interesting thing with Cas12 is, and I didn't tell you this because I didn't want to go too much in experimental detail, is that there's a big difference between Cas9 and Cas12. If it comes to the cutting side of Cas9 is defined in the most conserved, called also seed part, where you have a 100% homology is necessary, or base pairing is necessary for cutting. And this is different with Cas12a. It's cutting away from the seed sequence, so there might be some changes tolerated. And this, in principle, enables that certain junctions might be cut several times. And if you have several times the choices between homologous recombination or end joining, the homologous recombination adds up over time, and that's why we hope to have more homologous recombination. And here's the control for this site, which we already used then later on for gene targeting. Now this is end joining, and here you see LB-Cas12. This is the original enzyme when we performed this experiment, not the improved. The improved we only developed later. You see we have less cutting than with aureus Cas9, but if it comes to gene targeting, it's the opposite. So the blue bars prevail here in comparison to the red bars. And so all in all, we think that LB-Cas12 is 3 times better for inducing gene targeting than aureus Cas9, and we again have at least some lines where we have higher percentage rates. And we also tested later on than TT-LB-Cas12 in a direct comparison with the original LB-Cas12, and again, as expected, TT-LB-Cas12 is the best enzyme, not only for inducing mutation in plants in our hands, at least in Arabidopsis, but also for performing gene targeting experiments. So if you're interested to performing gene targeting experiments, my take-home message would be, please use the temperature optimized LB-Cas12. I think this is the enzyme to use. This is the best enzyme. Okay, and now I come to the third part of my presentation, and this is in principle now the chromosome engineering, and I want to show you some results there. So in principle, you might ask, why is chromosome engineering interesting at all for breeding, and what is chromosome engineering all about? So there are 3 simple ways. If you have 2 breaks to make chromosome engineering, you can make a deletion. And we showed already all these years ago that this can be efficiently done in plants by inducing 2 breaks. But neither if you get an inversion nor the exchange of chromosome arms was shown till now. But both, let me state this clearly, both are very interesting if it comes to breeding. Why? Because, for example, with translocations, you can easily break genetic linkages. Often you have the problem in breeding that you have a trait that is a good trait directly in close proximity to a trait that you want to get rid of. So good taste with a virus sensitivity, for example. And if you now would be able to part these two, you could get rid of the virus sensitivity. And the way to do this is to just get these apart by a translocation. So you cut here and you cut here, and then you have one gene on this chromosome, one gene on this chromosome, and then you can segregate the bad trait away from the good trait. And inversions are also very important because nowadays, using long sequence read, it turns out that there are a lot of inversions occurred during evolution between wild species, wild closely relatives, and cultivated plant species. And in all these regions where there are inversions, there is no genetic exchange possible. So if we could revert an inversion, then we could also induce genetic exchange in this region. And the most researched inversion in plant biology is, of course, an inversion in Arabidopsis. This is the 1.2 megabase NOP sequence. And here you see it's on chromosome 4. And if you cross different cultivars, some cultivars of Arabidopsis have this inversion, other cultivars don't have this inversion. If you cross the two, where one has the inversion, the other one doesn't have the inversion, you see here is the genetic map. There is this region where the inversion is. There is no recombination at all. No crossovers occur in this region. It's absolutely dead. And of course, we ask ourselves, are we able to induce inversions? And this is work done by Carla Schmidt, another very motivated PhD student who just left the lab. And in the beginning, we were not so optimistic that we really can inverse bigger sequences. So we started with 2 KB sequences on different chromosomes. We analyzed this via digital PCR and just ask ourselves, are we able to induce inversions? At what frequencies are we able to induce inversions? And what you see here, if you induce 2 breaks, it depends a little bit on the locus and on the cutting efficiency. In about 1% of the cases, you should be able to induce an inversion. And so we, of course, then analyzed these new junctions that were formed. So in principle, if you make an inversion, 2 new sequences are joined. And of course, you can then determine the junction. And what we found out, and this was at this moment quite surprising for us, was that indeed most of the junctions were perfect. So if you think about CRISPR-Cas inducing mutations by a single break, you also see deletions or insertions or whatsoever, sequence change. But if you bring in these 2 sequences that in principle cannot be recognized anymore by a guide, because the guides are only cutting the other sides, but not the newly formed junction, apparently then the sequence is conserved. And this, of course, indicates that in general, most probably there are several rounds of cutting occurring also for all break sites till a mutation is induced, and then it's of course stabilized because it cannot be cut anymore. And we were in principle interested to find this, and then we thought back, and I mentioned these results in the beginning, that there are several end-joining pathways, and this is typical for the classical end-joining pathways, that the ends are conserved. And you see here, the classical end-joining pathway is shown here again, and this is a microhomology-mediated pathway. And in the classical pathway, you have enzymes that protect the individual ends. This is the so-called Q heterodimer. And the Q heterodimer, in principle, protects the ends. And therefore, we ask ourselves, what would happen if we use knockout mutants of these genes? And we expected, of course, that inversion frequencies would go down. But what we found is that inversion frequencies went up. And this was very surprising for us in the beginning. But it turns out that the safeguarding of the ends, that no degradation is taking place of Q70, does not only apply for each end, but that these heterodimers keep both ends also together. So in principle, if you get rid of these DNA binding blocks, then the 2 ends fall apart. And then the other pathway, can take over and the other pathway doesn't care what kind of ends are joined. And therefore, we see in principle more events after we knocking out the classical end joining pathway. And in principle, this is of course also a possibility to enhance the frequencies of bigger rearrangements, if you're interested to induce bigger rearrangements in plants. The downside, if you do this, is that the junctions, because they are not protected anymore, are not as conserved as before. There are a lot of deletions going on. Here you see this comparison, much longer deletions than with wild-type plants. And so in principle, after showing that it's possible, we then also tested, can we get heritable events? And we used again the same setup as before for gene targeting with the XL promoter expression of aureus Cas9, and we find a number of lines where we really get T2 inversion frequencies of up to 10%. And this was so encouraging for us that we said, okay, now let's go for the big prize. Let's try not only to inverse 2 kb, but to inverse 1.2 megabase pairs. Can we achieve this? And we started these experiments. And indeed, Carla was able to find this new junctions that indicated that we get the inversions, and we got quite a reasonable number in different lines. Of course, you have to screen, and you screen, I don't know how familiar you are with Arabidopsis, but you have to screen, of course, a few bulks of T2 plants, and then you go for the ones where you have a positive signal, and then you screen individuals. But finally, Carla was really able to obtain these plants. And of course, it's very important to have the junctions, but in principle, you also have to show that you made an inversion. And how can you do this? Well, we did this together with Paul Franz, who originally discovered this inversion and characterized this inversion, and he characterized it by using back clones in a cytological analysis, where he can really mark the individual parts of a chromosome one after the other. And what you see here in wild-type plants, you should have the order red, green, red, and this is shown here. And with our inversions, we have red, red, green. Isn't it nice? So in principle, we really were able to induce the inversion. And as I told you, what is important in the end is not only to inverse this, but to show that then we are able to induce meiotic recombination in a region that was dead for 5,000 years. This is how old the inversion was in this case. And indeed, this was done with my good friend Andreas Huben. We could show, here is the restored line, the reversed line. We could show recombination here. These are two different ways to measure recombination, either in pollen or in the next generation. And we could really bring back recombination crossovers in this specific region. Okay, so in principle, now we can inverse sequences in chromosomes. How about translocation? This was work done by another PhD student, Natalia Beijing, in my group, and she tried to exchange the ends of the chromosome arms 1 and 2 of Arabidopsis. And the experiments were prepared also using first digital PCR. And we also tested, because we knew this already, that the Q mutant might be very interesting. And here you see again this effect. In general, inversions are more frequent than translocation. Translocations are more seldom than inversions, but also the translocation frequency you can enhance by using the Q mutant so that the ends are not protected anymore, not kept together anymore. So this might be a nice trick in general to get more rearrangements, also more translocations. And then we set up the same system we used for the inversions with the aureus Cas9 and the XL specific expression, screening a big number of T2 pools. And as you see here also, Natalia was able to get positive individuals where we got the right PCR junctions, as well in wild type as in the Q mutant. And here are the respective junctions. And what is really fascinating for me is that you exchange chromosome arms between chromosomes, and many of the junctions are 100% conserved. So you change chromosome arms without using a single nucleotide. So this is really fascinating. And what is even more fascinating, to see this then cytological, this was done again by Andreas Huben, our good friend. And here you see the two chromosomes before, and here you see them after the translocation. And this is possible also for other chromosomes. We then also induced a translocation between chromosome 1 and chromosome 5 of Arabidopsis. Here is the junctions. We didn't work with tremendous amount of plants, just to show the principle. And indeed, what you can see here again are these fused new chromosomes. Isn't it nice? So these are the original chromosomes, and here are the chimeric new chromosomes we generated with CRISPR-Cas. And so in principle, now the door is open for using chromosome engineering in plants, and this will allow us to have new means to solve old problems of breaking and fixing genetic linkages. And there was just a publication also in Nature Plants, where people could show that a large inversion is also possible in corn. And I think that we have now in hand also much more options that we can use with this technology. We can restructure genome evolution. You know that the chromosome numbers, the chromosome structure changed many times during plant evolution. We can now mimic these events, learn more about evolution, but we also can, in principle, now create synthetic plant chromosomes. We can change the numbers of plant species, and in the end, we might end up in creating new plant species that from the primary sequence will not look different from their progeny, but still you would not be able to cross them to these original plants anymore. So there's many, many different interesting aspects you can think of that we are all now have in our hand, where we can, in principle, use the technology for breeding, but all as well as establishing a new level of synthetic genomes in plants. And with this, I'm at the end, and I want to thank you for being so patient and listening to me for 3/4 of an hour. This is my group. Obviously, this is a picture that was taken before the CRISPR-Cas time. And I also want to thank, as it was mentioned before, the European Research Council for very generous funding my group, my research on CRISPR-Cas for the last 8 years, and there will be 2 years more of funding. This really made it possible for us to establish this chromosome engineering technology. Without this money, we would not have been able to do this. And with this, I want to thank you for your attention. And now I think I'm finished and ready for questions.
Thank you, Dr. Holger, for this amazing and interesting presentation. Now, as you said, it's time for the questions from our audience. We would like to remind you all that your questions have our undivided attention on Zoom, YouTube, and Facebook. If you are on Zoom, you can enable your microphone and directly ask Dr. Holger, or you can also write down your questions on the chat and I'll read them to our guest. So, Gustavo Rosoto asked, could it be possible to ask for his email address?
Yes, I mean, you just go to Google and give in my name, and then you will find my email address and also my homepage at the Karlsruhe Institute of Technology. And there's also a lot of information on our material, but also there are several YouTube videos with talks of me and publications. And you can also see all our publications and load them down, whatever you're interested in. And if you want to get the clones, just write me an email. Yes, of course.
Thank you, Dr. Natalia says, thank you very much for the wonderful talk. Very enriching for all of us.
Hello, Doctor. I have one question. You say that at the start of your presentation that the usual or like the quotidian or mutations were running out. So in this sense, CRISPR-Cas will like change that this present situation to have like more mutation available that will help to the 2050, 50, I think, right? For scrapping problems on what do you think how CRISPR can help agriculture and all these problems seen in 4
So
years?
there are of course two different developments. The first development is that due to our research over the last, as plant biologists, over the last 50 years, we know a lot about gene functions. So there are a lot of genes that are, for example, involved in pathogen response. So the pathogens have to always find a molecule where they can dock on, like for example, fungi. And if you knock out this molecule, you are able to induce resistance in fungi. This is an approach that has been used in wheat, for example, to get the fungi resistance. On the other side, besides the single traits, we might also have a chance to induce a row of mutations at the same time. And my good friend Dan Wouters has shown in a fantastic paper about 2 years ago, 1.5 years ago, that you can domesticate wild plant species by inducing a certain number of mutations that in principle is a reproduction of what happened to classical domestication over years with specific genes. And he could use this technology to produce from a wild tomato a cultivated tomato. And Zach Lippmann did the same with eggplants. He produced eggplants with much bigger fruits. But if you think about this, we will be able, using this kinds of technology, also to take wild species that are resistant to heat and salt stress and make out of this species better crops. So in principle, we don't have in any case to bring heat and salt stress in our cultivated crops, but maybe it's easier to just take wild species like teff in Africa and make out of this plant a cultivated plant by changing fruit size, protein content, things like this. I think this makes me very enthusiastic. And these are really game changers also in our way how we can handle the current situation of global warming. And that's why I think it's so important that we all over the globe use this technology. And that's why I'm personally so disappointed that in Europe we have this horrible problems that the technology is accepted by certain politicians.
Yeah, it's actually a big problem all over the world, but I think that is going to change in the next 5 to 10 years. Thank you, Doctor.
Any other question? Remember, the chat is completely open if you have any question. Well, seems like everything is so clear. So now for the closing, I would like to ask to the audience to turn on the cameras to take a picture and for this moment. ¿Algunos pudieran encender sus cámaras, por favor, para poder sacar la foto? So now 1, 2, and so finally, we would like to remind you that tomorrow we will have our last CRISPR talk. So I would like to see you everyone tomorrow, then with another specialist in this interesting technology. Thank you, Dr. Holger. Thank you.
It was a pleasure. Thank you.
Thank you. Yeah, thank you for all the audience. So for your assistance. So see you very soon.
Bye-bye.
Bye-bye.